Good morning, good afternoon, and good evening, everyone, and welcome to our fourth event in the 2023 Neurophysiology series titled The Impact of Sleep and Circadian Disruption on Human Health and Disease Risk, featuring Dr. Josiane Broussard, Associate Professor in Health and Exercise Science at Colorado State University. Today, she will present her research on the links between sleep, circadian disruption, cardiovascular function, and metabolic health. We'd first like to acknowledge our partners for this series, the American Physiological Society, the Society for Neuroscience, the Federation of European Neuroscience Societies, the American Autonomic Society, and Scientist.com. We'd also like to say a big thank you to all our series sponsors for helping make this fantastic program possible. And with that, Josie, I'll hand it over to you whenever you're ready.
Awesome. Thank you so much, Andrew. Just also want to give another shout out to the APS for organizing these incredible webinars. I know my lab takes advantage of them, so I think it's a really wonderful resource. And just like Andrew said, please check out where your polls are, because I have a couple as we get started. So thanks again for joining me today and to hear a bit more about the research in my lab and in Colorado on sleep and circadian rhythms. So with that, the first thing I like to do is really thank my lab. And as many of you may know, sleep and circadian research, especially in humans, but actually probably any type of research in sleep and circadian rhythms, takes quite a small army of people, participants, students, trainees, to really help us run these studies, especially because in my lab, we host participants in the lab for multiple days, and we monitor them for many days at a time, 24/7. So it takes a big group. And so we gave up a long time ago trying to take a group photograph. So now we place people in funny positions around the world, and then we Photoshop ourselves together. So this is a big shout out to my lab. And also, so I don't run out of time at the end, I just want to acknowledge our funders. So the lab has been very fortunate in obtaining funding over the years. And I also want to just give a shout out also to any place, in this particular case, University of Colorado and Colorado State University, but really any place that provides junior faculty funding is a wonderful place to be, because that can really give you practice and experience putting together smaller awards as you build up to your kind of bigger NIH level awards. So on to some science. Why do we sleep? Well, this is the reason why I'm so interested in sleep research, is this XKCD comic, which is that we spend so much time doing this behavior about which we know very little. And so this was really what got me interested in sleep research in my early years, and what we're going to talk about a bit more today. So I mentioned, what is the function of sleep? We really don't know. I mean, we know sleep is restorative, and as you'll see in the next slide, we also know that animals will die without sleep, so total sleep deprivation is fatal. But how it performs this restorative function is totally unclear, unknown. And if you think about that from a real existential level, I mean, are there any other behaviors that we do that are fairly involuntary about which we know so little? And I would argue that there's nothing like that. We know quite a bit about physiology and why things do what they do in the human body, but we don't know a lot about sleep. So that continues to hold my interest and curiosity. So as I alluded to in that last slide, total sleep deprivation is fatal in rats. So this was work that was done in the mid-90s at the University of Chicago, which is where I ended up going for my PhD. But they characterized this model of sleep deprivation in rats using the disk over water, which we know a lot about now. It actually is quite stressful. But at the time, using it for total sleep deprivation led to the animals becoming disheveled and scrawny. They got skin lesions. They overate, they had hyperphagia, even in the face of severe weight loss and body fat loss. They lost the ability to thermoregulate, and they all died. So this is a quote from the paper, that no rat survived unrelenting deprivation of sleep. So also, as I mentioned, all animals sleep. Mostly I just show this because I'm like the luckiest researcher in the world, because I can Google cute animal sleeping image search and put anything I think is cute in the slides. So actually, if you have any cute animal pictures, you can send them to me. And all of my friends and family know if they fall asleep in front of me, they are probably going to get put into a talk. Okay, so all animals sleep. And actually, so hopefully you saw this figure that just popped up, which is jellyfish, Cassiopeia. And about 5 or 6 years ago, there was a paper in Current Biology that reported on this particular jellyfish, and it reported a sleep-like behavior. And that doesn't sound super big to you maybe right now, but this was the very first time that a sleep or sleep-like behavior was reported in an organism without a brain or centralized nervous system. And so this was huge, because up until that point, there was kind of this concept in the sleep field that sleep is performed by the brain. The brain is really doing the function of sleep. And you must have a centralized a brain or centralized nervous system to perform this behavior. So this was a pretty big deal. And there's sort of I would love to someday meet the students who figured this out, but really it was supposedly kind of some night science, where students were in the lab, like tapping the tank at night, and they sort of woke up the jellyfish, and the jellyfish would go back to sleep. So that's a shout out to students, is that if you are curious about something, there's this thing I call night science that you can do, that maybe your PI will find out about later when they get a high impact paper. Okay. So what about tissues? So not only do all these animals sleep, but almost all cells in the body also possess the core circadian clock components. So the small molecules that are coordinated to produce rhythms in all tissues thus far examined, or I should say most tissues. So we know that the central clock is located in the brain and can project out to peripheral tissues and synchronize all of these other clocks in the body. And why that's important to think about from an evolutionary perspective is all of those organisms and cells and tissues evolved when the night sky looked like this. So when there was a really distinct day and night cycles, which we know, of course, is no longer the case. And these are some satellite images of light around the world. We know now that we can manipulate our environment like never before. So we have constant communication, commerce, travel across time zones, things like that. And we can flip on the lights like we never could at any other time in human history. So that has in part played a role in what I'll show you in this graph, which is the change in sleep duration over the last 50 to 75 years. So these are US data. So these are from self-reported sleep questionnaires. And you can see that over this time period, average sleep duration in the US has gone down by about 2 hours, a little under 2 hours. And this is, like I said, these are US data, but this is considered a worldwide phenomenon, that sleep duration has gone down. And in that same time period, we know that obesity and the metabolic syndrome have gone up. So really, the bulk of my research has focused on this link between sleep and circadian disruption and the metabolic syndrome. And I'll point out this paper by Spiegel et al. In 1999 was the paper that launched the field. And so the senior author from that paper is my PhD mentor, Dr. Ev Van Cotter, who was really one of the first scientists to ask, great, sleep is happening in the brain, but what does sleep do? What is the impact of sleep in the whole body, like all the peripheral tissues. And so this was the very first paper that reported on that and really launched my field. So this is a meta-analysis, so results from a meta-analysis that I'll show you, which is looking at type 2 diabetes risk and traditional factors. So if you see, as I overlay the factors here, these are the 3 things, if you're going to your primary care doc, they might ask you about or check for, being overweight, a family history of diabetes, and physical inactivity. These are considered the 3 big risk factors for type 2 diabetes. Now, if you watch this title slide rotate, what this meta-analysis did was overlaid sleep and circadian factors onto these top 3 to see, what is the link between type 2 diabetes risk and these factors. So you can see, and you can guess based on where the spacing is, all of these different ways of assessing sleep quality and circadian factors were associated with an increased risk for type 2 diabetes. So this odds ratio is higher than 1. So anything past to the right of the dotted line was indicative of an increased risk for type 2 diabetes. So if we look over on the left, let's see, obstructive sleep apnea, difficulty maintaining sleep, shift work, initiating sleep, so kind of forms of short sleep duration, and reduced sleep quality, things like this. So we can see all of these black boxes are associated with this increased risk for type 2 diabetes that are comparable to some of the big traditional risk factors. So this really highlights the clinical significance of sleep and circadian disruptors, because it's not just a non-clinically relevant risk factor, right? Like these are actually as important as some of our traditional risk factors for type 2 diabetes. So for the rest of the hour, is that about the time? I will go through some of our work, and here's the outline here. So I'll speak about our work in humans. Also, for the trainees out there, I'm going to tell you a little story about my segue into large animal research for my postdoc, and then tell you a bit about our current work and future directions. So sleep and circadian disruption in humans. So as I mentioned, all of these tissues possess the core circadian clock machinery. And so historically, like my research has been in clinical for the most part, but really with this interest in tissue specific impacts of sleep and circadian disruption. So where does some of that research get done? This is our sleep lab. So I just wanted to show this beautiful sleep lab at Colorado State University. But I want to point out to the trainees that my sleep lab at CSU was built based on all the sleep labs I've ever had the fortune to work in. So based on the sleep lab at Harvard Medical School, where my very first mentor, Janet Mullington, worked and first got me interested in sleep research. Then the University of Chicago with Dr. Van Cotter and Dr. Tasali. Then some animal work in Los Angeles. And then my circadian mentor, Ken Wright, here in Colorado at the University of Colorado at Boulder. So using all of those beautiful examples, we actually had the sleep lab built at CSU. So it's set up to look a bit more comfortable, a little more cozy, like a hotel room or a dorm room, but we still have all the clinical capabilities. So this is a hospital bed. Participants, as I mentioned, can live in the lab for any number of days. Right now we have a study in which participants live for 6 days in the lab. And they can work at the desk. They can store their stuff in this little bureau under the TV. But the most important part of this room is on the left side. I'm not positive you can see my mouse, although I keep using it. Is not a light switch, but a through the wall blood sampling port. And this, as you'll see, becomes a really important aspect of our work because we want to know a lot about what is changing in blood and the circulation overnight, but we want to know that without disturbing sleep. So what we can do then is we can connect the IV from the participant. We can actually connect it to a long line, feed that through the wall, and we can very gently sample blood from outside the room while participants are asleep. And we can do that with limited disruptions to sleep. Okay, so for the first study I'll talk to you about, this was conducted at the University of Chicago, as I mentioned, with my mentors, Dr. Van Cotter, Tasali, and Brady. And this study was a insufficient sleep protocol. So it was an inpatient study. And this is a protocol schematic of the normal sleep condition on the left. So each night is represented by a single line, and the white box indicate the sleep opportunity. So I'll show you data from 24-hour sampling, which is shown by the yellow dotted line here, as well as data from an insulin sensitivity assessment, the IVGTT in the yellow line, and fat biopsy. So these same participants came on another visit, separated by at least 1 month. And as I mentioned, this was randomized crossover design, so participants could have started with the insufficient sleep condition. So you can see on the right side in red, the participants, the same people came in and they spent another week in the lab. But in this condition, the first 4 nights were short sleep to 4.5 hours time in bed, followed by recovery sleep. But the outcome assessments were the same. So for insulin sensitivity, so here we see an insulin sensitivity index, and this paper was actually published by my postdoctoral mentor. And just a quick note about insulin sensitivity and changes to insulin sensitivity are a really important risk factor for future development of diabetes. So you can see across the y-axis here, different demographics of people. This diabetes, non-insulin dependent diabetes mellitus, is at the very bottom. So you can see healthy men and women have very high insulin sensitivities. And as you go down the spectrum, you can see then people with diabetes have quite low insulin sensitivity. So our study, and I'm showing now actually a combination of results from many different studies, which have been very consistent, that when people are sleeping at their healthy habitual time, which in this case is between 8 and 9 hours time in bed, their insulin sensitivity is very high. So they're falling between very high insulin sensitivity groups. But when those same exact people come in and spend 4 nights in the lab where they get insufficient sleep, their insulin sensitivity goes down significantly. And they now fall between 2 groups at known higher risk for diabetes. And this again reinforces that clinical relevance of insufficient sleep. You know, it's not a small amount that they are impaired when they perform insufficient sleep. And so we're taking healthy, young, normal people and essentially making them look pre-diabetic when they are in the lab not getting enough sleep. Okay, so a little more specifically, what does glucose look like? So in these studies, we also measure interstitial glucose using a continuous glucose monitor. And here you can see this is now time since waking up across the x-axis, and interstitial glucose you can see across the y-axis. During normal sleep, the arrows indicate meals. So you can see in response to breakfast, lunch, dinner, and snack, you have these nice glucose excursions. When these same participants are undergoing insufficient sleep, we now have a much different glucose profile. There's a much larger glucose response to the first meal of the day. And you can see something probably going on during sleep as well, which if you have some ideas, shoot me an email after, because we have not really thought too much about this yet. These are unpublished data. So anyway, so you can see here, obviously very big changes in response to the meals. And it's important to note that these meals are actually identical. So from the breakfast meal in the white line compared to the breakfast meal in the red line, that is the same exact meal being given. Okay, what about free fatty acids? So and still am very interested in the impact of sleep and circadian disruption on adipose tissue, which release free fatty acids. So you can see here now across the x-axis are just the nighttime period. And you can see when participants sleep normally in the lab, there is a big nocturnal surge in free fatty acids, which has been characterized previously. But when participants are sleep restricted, you can see the same pulse, but a little bit widening of the nocturnal free fatty acid pulse. So a significant increase in the late night and early morning levels. And again, this isn't, I don't like to overstate this. It's not a huge drastic increase, but this is after 3 to 4 nights of insufficient sleep. So what must lipids and circulating fatty acids look like if these behaviors are continued more chronically? Okay, and I mentioned the adipose tissue, which is doing the thing of releasing the fat. What is going on there? So in this study, we also took a fat biopsy, as I mentioned in the protocol figure. And this is the method that was used. It's a small, kind of like mini liposuction, where you can aspirate a little bit of the subcutaneous adipose tissue. And for the results I'll show you, we focus on, this is a very simplified version of the insulin signaling cascade. So in response to insulin binding at the cell surface, a signaling cascade is initiated, which ultimately results in the translocation of this glucose receptor, GLUT4, to the surface of the cell. So for the results I'll show you, we focus on this central signaling molecule, AKT, which when it becomes phosphorylated, that's considered the active form that kind of propagates this message. So here I am, I'm not taking the biopsy, but this is my little hand, taking the fat cells back to the lab, washing them, keeping them warm, and then, stimulating them with increasing doses of insulin. So these are data from a Western blot. So this is a protein expression. Looking at normal sleep. This is now from one participant, these data. So you can see that phospho-AKT, so that's when the AKT becomes activated, you can see the insulin dose down at the bottom, and total AKT is used as a loading control for this type of experiment. And you can see when the AKT becomes initially phosphorylated and also when it's maximally activated. So when fat cells from that same participant are sleep restricted, you can see this very clear shift in both the insulin dose at which the AKT first becomes phosphorylated, as well as the dose at which it's maximally activated. So when we quantify that from all the participants in whom we took fat biopsies, we can see that down in this line graph. So again, normal sleep in the closed circles, again, as insulin concentration increases, as the dose goes up, we see an increase in the ratio of phospho to total AKT. But during sleep restriction, there's a right shift. So this is really indicating that the fat cells themselves became resistant, or at least less sensitive to insulin when they were sleep restricted. What about other metabolic tissues? So when we think about the big 3 metabolic tissues in the bodies, we typically think about adipose tissue, liver, and muscle. It's not so easy to get muscle tissue from humans. So I told you a little bit about fat, but as I've moved to Colorado, I've become very interested in muscle, because that's a lot of the expertise here in Colorado is focused on muscle biology. And so for this next study, I'll tell you a bit about that. And so this is the team here. I mentioned Dr. Ken Wright, my circadian mentor, Brian Bergman, and Ed Melanson are both at CU at the Anschutz Medical Campus, and were mentors on my K. Also Dr. Irene Schauer and Melanie Cree-Green, who are both muscle biopsy experts and endocrinologists, and of course my wonderful Sam lab manager. Shout out to her, who runs our lab, and she's the boss. I tell her she's my handler. and she keeps me in line. Okay, so the question is, we do circulating lipids. So I mentioned we see that increase in fatty acids. We know there's increased lipids in circulation with sleep loss. Do they accumulate and get in the muscle? That was our question for this next study. So quick cul-de-sac on muscle lipids. So these are some examples: triacylglycerides, diacylglycerides. So you can see here, this is an image of a triacylglyceride on the top. And then when one of those free fatty acids is hydrolyzed and released, we then can have diacylglycerides. And you have probably, I'm sure you out there are much more experts than me in muscle lipid species, but you can see here on the if you can't see my mouse, it's the left bottom lipid is a 1,2 diacylglycerol, 1,2 DAG. And on the right, because it's the fatty acid is in a different position, it's a 1,3 diacylglycerol. So experts out there certainly know that 1,2 DAGs have been heavily implicated in insulin resistance in muscle. So you can see here from where we take the muscle biopsy from the vastus lateralis muscle in the leg. So from our muscle biopsies, and with Brian Bergman as the primary mentor on this project, we looked at total triglycerides as well as saturated tags. We don't see any differences between normal sleep and insufficient sleep. But getting back to looking at those 1,2 DAGs, which have been previously implicated in insulin resistance, if we look at those, we can see, sure enough, that during habitual sleep, if you look at the white bars, compared to insufficient sleep, we do see a significant increase in some 1,2 DAG species. You can see the chain lengths down at the bottom during insufficient sleep. So we wanted to know, is it all DAGs? Is it just the 1,2? So here, if you watch my little blue circle slid over to the 1,3 DAGs. So that's a different position of the free fatty acids on the glycerol backbone. And they have historically not been implicated in inducing insulin resistance. So when we look at those, sure enough, we don't see any significant increases in 1,3 DAGs during insufficient sleep. And even in some cases, we see a reduction. Okay, so moving on for a quick story in the large animal model. So after my PhD, so if the trainees are out there and you're doing clinical research, there's a lot of recruitment involved, participants come late, maybe they no-show, and you're like, man, I need a break from all these humans. So that's sort of how I felt after my PhD. And I thought, I want to get some hands-on, really mechanistic training in metabolism. So I shifted gears and went to the dog model. And to do that, I spent a few years with Richard Bergman in Los Angeles. And he is, as many of you I'm sure know, a giant in the field of metabolic physiology, especially when working with the large animal model, the dog. And so he's really characterized the how fat feeding in the dog model induces insulin resistance and metabolic dysfunction. So I was really interested, as I mentioned, getting this hands-on training, working with an animal model, but because of my sleep and circadian background, I wanted to work with a diurnal mammal. So an animal that sleeps at night and is awake during the day, like humans, unlike the nocturnal mouse. So that's how I ended up in my postdoc. And so in this study, animals were undergoing high-fat diet as part of Dr. Bergman's primary interests. And so insulin sensitivity was assessed. So you can see here in the white bar, this is normal sleep, animals just living regularly in the vivarium on a controlled diet. And you can see they have, again, that high insulin sensitivity considered very healthy. And during a high-fat diet, as I mentioned, this very well-characterized model of high-fat feeding, you can see a significant decrease in insulin sensitivity after this was about 6 to 9 months of a high-fat diet. So again, introducing a little bit of night science of my own, I convinced Dr. Bergman to let me do a 1-night total sleep deprivation study in these animals before they went on a high-fat diet, so that we could actually compare sleep deprivation, normal chow feeding, to the impact of a high-fat diet alone. And so I did this in a random order, so it's not necessarily the order that I'm showing it here. And you can see now the blue bar is insulin sensitivity after a single night of total sleep deprivation on a chow diet. So now you can see how comparable that is to high-fat diet. So this was quite striking to us at the time. And again, it's a transient impairment, because the animals do go back to normal, which we know because I did, like, as I mentioned, I did the sleep deprivation versus normal sleep in a randomized order. But again, the level of comparability was really striking and exciting to us. So I often think about, and you can ask me about this at the end, what can change so quickly that we see such an impairment after only a single night? Okay, so back to humans. So, the dog model is really a wonderful model, but I did feel compelled and responsible, really, to do work in humans that can be done in humans as opposed to animals. So I had always planned to go back to human research, and that's how I ended up in Colorado. So I'll talk a bit for the next couple of minutes about circadian misalignment. And what do we mean by that? So we define that, or at least Ken and I define that, as an altered or inappropriate relationship between sleep-wake and associated behaviors. So what does that mean? When we think about sleep-wake and associated behaviors, we think, for example, awake being associated with eating and being active, and also all of this occurring in the biological day when the sun is up, versus being asleep, fasting, and inactive during the night when it's dark out. So any mismatch of these behaviors we would call circadian misalignment. Even if you, let's say, woke up early to catch a flight and ate during what is still your biological night, or even just being awake during that biological night, we would actually call that a form of circadian misalignment, morning circadian misalignment. So why is circadian misalignment important to think about? Well, we also know that shift workers are 40% more likely to have type 2 diabetes. And this appears to be a dose-response relationship. So the risk for type 2 diabetes increases with the number of nights worked per month. And this is not a small population of the US employees or the world, where we have 1 in 5, and actually now I think the report is over 20%, closer to 25% of workers work what's considered non-standard hours.
Okay, so here you can see the protocol. So this is what we would call a slam shift protocol. I'll try not to repeat exactly what I said, but here we can see participants stay in the lab, and we essentially make them look like night shift workers. We simulate night shift work in the lab. So participants have 2 nights of habitual nocturnal sleep, followed by a transition day, which is common in night shift work, where participants might try to get a nap in the afternoon, followed by an overnight shift, and then 2 nights of daytime sleep. So I'll show you a bit of data from the red dotted lines on day 2 and day 4, and I'll refer to those as circadian alignment and misalignment. So here again, we see glucose, which I showed you before, but this is now plasma glucose from the blood. And across the x-axis is time since waking up. So you can see here, the yellow arrows are meals. So again, we see that nice meal response during circadian alignment. And then we see a very consistent pattern with circadian misalignment, as I showed you before with insufficient sleep, where that first response, that meal response to the first meal of the day, is significantly higher during misalignment. And one thing to note here is this meal is actually eaten in the afternoon during day 4 circadian misalignment, because participants wake up probably around like a relative clock hour of like 5 p.m. Okay, what about my favorite circulating factor, free fatty acids? So here you can see, last time I only showed you the nighttime period, but this is free fatty acids across the day. Free fatty acids release or lipolysis is suppressed by meals. So you can see following the meal, there's a suppression in free fatty acids. And this is white is circadian alignment, and now red circadian misalignment. We see that the free fatty acids are significantly higher, especially when participants are waking up and eating during the biological night. Okay, so this is, we come to the final like direction of my talk, which is actually future directions and ongoing studies. But here, what I'm going to let you do is actually choose your own adventure. So this is our final poll. And Andrew's bringing that up now. And so now the question is, with our last few minutes, what would you like to hear about? So as you're thinking, I'll tell you, everyone is wondering, especially if you're an exerciser, I see you, because you're wondering, what if I get up early and I don't get enough sleep because I want to go on an early morning run? So I get that question a lot. So we've actually done that study. Another question I often get is, what about people who don't get enough sleep? Can we pay back that sleep debt? Or people with, health impairments? What if we get more sleep? Can we improve things? And then a future direction and ongoing study is, what about changing the timing of behaviors? What about the time when we sleep or the time when we eat? And what might that mean? So let's see. Oh yeah, and then a couple other existential questions that you could also submit. Okay, so I'm gonna see, what does the poll say? What if I wake up early to exercise? Okay, I'll give you a little more seconds. But I think that checks out, right, with who you are as APS members. So it's looking like, what if I wake up early to exercise? Okay. Andrew, can you just let me know if you're seeing everything again, since I went out to look at the poll?
Yes, everything's perfect. Yep.
Okay, awesome. Okay, so I built a little wormhole into the slides depending on your answer. So what if I wake up to exercise? So I'll tell you a little vignette about that study, which is also which is unpublished, which I should be submitting to my own call that Andrew mentioned, so that I have 2 calls out for papers. But to be honest, I probably won't get my act together in time, because it's actually taken more time to manage the call than I've been able to write my own paper. Okay, exercise during sleep loss. So in this particular study, we recruited 22 young, healthy participants. We put them on a very strict schedule for 1 week prior to coming into the study, again, based on their habitual times. They were given an energy balanced diet prior to the study, and they weren't allowed to exercise the day prior. And what we put them through was an insufficient sleep with and without exercise protocol. And so on the left, again, this is kind of, now you've seen this a number of times, this is the inpatient protocol with the blue boxes indicating the sleep opportunity. So after the first baseline night, participants were delayed in their bedtime by 4 hours. So they were getting 5 hours time in bed, but waking up at the same time. And then we performed a glucose tolerance test. And in the other half of the group, so we did the exact same thing. They had the same insufficient sleep schedules, the same glucose tolerance test, but in those participants, they performed daily exercise in the lab. And all else was matched. So even the room where participants went to exercise, if they were in that group, we actually had the non-exercisers go to that same room, even just so that we would have light control. And instead of exercising, they had a cozy armchair and read a book. So what happens to insulin sensitivity? So this is now a different test, so not the IV glucose tolerance test, but an oral glucose tolerance test. But we see during when the people who were sedentary, so shown in the white bars, we saw a significant reduction in the OGTT Matsuda index of insulin sensitivity, which is again consistent with all these other studies I showed you. But then when a group who came in and did physical activity, which is the black bar, you can see they still had a significant impairment, but the impairment was, there was a trend for them being different. So it was somewhat like partially mitigated the impact of sleep loss on insulin sensitivity. So just to give you a little more raw data on that, these are the glucose and insulin curves in response to the oral glucose tolerance test, which involves participants drinking a sugary drink. So you can see this in the sedentary group, the glucose response to the drink and the insulin response to the drink at the bottom. When those same participants, sorry, it kind of overlaps my legend, but then the dotted line is when participants are getting insufficient sleep. So you can see the glucose is higher and the insulin is slightly higher. Now in the exercising group, you can see during habitual sleep there in white, and then insufficient sleep in the dotted lines. So there's clearly some differences going on that becomes more clear when we actually look at these profiles. So what does that mean for circadian rhythms? So now I'll show you some melatonin data, which we use. We take melatonin in the lab either from blood or from saliva in dim light conditions. And in this particular case, we took samples immediately when the participant woke up, and then every hour for 5 hours as a readout of the central circadian clock. So we're really curious whether the sleep plus or minus exercise impacted the central circadian clock. So this, now the white line is habitual sleep over that time period, looking at melatonin. And this is exactly what we would expect because melatonin should be low when you wake up. And you can see here, this is the tail end of the melatonin offset. And the little caret that just arrived indicates a very specific point in the melatonin offset curve, which we call dim light melatonin offset. And we use that as a circadian phase marker. So when those same participants are not getting enough sleep, there is a shift. Not only you can see that visually, obviously in the tracing, but we also have a shift in what is called the dim light melatonin offset, which is the time at which melatonin drops below a sort of field determined outcome. In this case, it's 3 picograms per ml in salivary melatonin. And you can see what this means is that participants experienced an almost 2-hour circadian phase delay. So just going to bed later at night for 3 nights led to this shift in the central circadian clock. And then if we get really nerdy, we can actually look at whether that shift, whether the size of that shift was related to the change in insulin sensitivity. And indeed, you can see the white dots are habitual sleep and the red are the same participants during insufficient sleep. So sure enough, we see the larger the delay, the bigger the impairment. So the more circadian shift we see, the bigger impairment we see in insulin sensitivity. So the very last slide is just looking at this in a slightly different way. This is now, again, the dim light melatonin offset, so the phase of the central clock by exercise group. So you can see here the offset during normal sleep in those sedentary people. They experienced a big shift in the circadian clock during insufficient sleep. So they went from, 1.25 hours all the way to almost 3.5. The exercisers who started off at the same circadian time as the sedentary participants, when they underwent insufficient sleep, they had a much smaller circadian shift. So we are diving into that with some work looking a little bit more specifically at the timing of exercise, because we do think that the timing of exercise we chose likely mitigated some of that circadian shift. So to leave time for questions, we won't be able to do any more of the adventures. So with that, I'll conclude. So hopefully I've convinced you that sleep and circadian disruption impair glucose and fatty acid metabolism, maybe as bad as a poor diet, and that's what we saw in the animal work. Even exercise isn't powerful enough to fully protect us. So that was what I really want to be the takeaway from that exercise study is we know that exercise is the most insulin sensitizing thing that we could do. It's more effective at improving insulin sensitivity than any medication out there. Even exercise wasn't enough to completely mitigate the effects of sleep loss. And this is a little bit of TBD because we didn't get to this particular future directions, but we may, maybe there's a way to optimize sleep duration and circadian timing of different behaviors, activity I mentioned, sleep, things like that, that could benefit metabolic homeostasis. And we're really interested in looking at that in free living shift workers. So with that, I will end with a few minutes for questions. Thank you.
All right, Josie. Yes, thank you very much for that fantastic presentation. We've got quite a few questions coming in. Thank you to everyone. Don't forget you can upvote them as well if any that you're particularly interested in hearing an answer to. And let's ready to jump in, Josie.
Sure.
Great. First question here. Is there any kind of link between circadian rhythm and cortisol level among the population?
So that's a great question. So we actually do, so cortisol is considered one of the kind of big three readouts of the core of the central circadian clock. So we do actually measure cortisol in the lab. There is a big spike in cortisol at wake time. And I think the research suggests that that cortisol is elevated certainly in the evening when people are sleep restricted. That was some work that came out of the Van Cotter lab. What happens to cortisol during circadian misalignment? Cortisol, like I mentioned, it does kind of follow the central clock. And it's impacted very, very strongly by waking. So if all behaviors aren't necessarily controlled for, cortisol can be difficult to interpret because it's very sensitive to changes in sleep wake, activity. So I guess I would sort of leave it at that and say that there's definitely a big connection between circadian rhythms and cortisol, but it just sort of depends on the question, I guess.
Okay, fantastic. Moving right along, I think you addressed this to some extent, but with acute changes in sleep initiation and or duration, are there changes in hormone expression, CRH, cortisol, adiponectin, and does it directly correspond to circulating glucose and free fatty acids, I believe?
Ooh, good. I mean, so good question. That's from someone who's like a hardcore physiologist, which I appreciate because some of the chapters that my PhD mentor Ev Van Cotter has published, that might even be one of the handouts of a chapter that we put out, aligns all of those hormones and all of those circulating factors. So I almost can't even answer that question because of course they're certainly all interrelated. And so I would actually point you, if you're interested in the 24-hour profiles and impact of sleep on all those different hormones, to one of those chapters. I think Hanlon is the first author on that sleep and endocrinology chapter that Andrew has as part of the handouts. But yeah, it's actually quite beautiful to look at, 8 circulating factors stacked, top to bottom, just to look at all the different rhythms. But certainly they're all interacting.
Okay, fantastic. What about late hour exercise? And the example here is 11 p.m. to 12:30 a.m. How does this affect sleep loss? The influence of that on insulin sensitivity, free fatty acids, and just general thoughts on exercise timing.
Great question. There's so much to do and needed, I think, for understanding the impact of different timing of exercise. In fact, there's a big RFA right now to specifically kind of look at that. And so you know, there's an idea and supporting literature that there's a phase response curve to exercise, which is, again, kind of some hardcore circadian biology talk, which is that there might be a time of exercise that shifts the clock in one direction or another. And the idea, the literature, the research would support that afternoon exercise will actually phase advance the clock, so kind of keep you to an earlier time, whereas other times of day might shift you in a different direction. Specifically, late night exercise is considered potentially disruptive to sleep quality, also because it can increase your core body temperature right before trying to sleep, which is exactly when your core body temperature you'd want to be kind of going down and hitting its nadir overnight. So that's generally, we would consider like exercise you want to have completed many hours before you're trying to attempt sleep.
Okay, fantastic. An interesting question here. It's a bit do you have time to continue as well here, Josie, a bit over the
audience? Totally.
Okay, fantastic. So thank you to everyone in the audience with the technical glitch. We'll carry on a little bit longer here, a bit over the time. Speculative question for you. What about circadian regulation of sleep and other functions in people who are blind?
Ooh, that's a cool one. I mean, that gets into some cool like existential stuff about, yeah. So it sort of depends the level of blindness. So there are different injury levels, or depending on, you know, the mechanism behind the blindness, where you actually still have light perception. And so participants that retain light perception will actually maintain normal circadian rhythm, similar to someone with full sight capabilities. If participants who don't get any light input, almost would be kind of akin to some studies that were done many years ago, where they actually brought participants into caves. And that was some of the really early circadian work, is where if you have no light exposure, you then will free run. So that means that you're living on your own individual genetically determined circadian period, which on average is a little bit longer than 24 hours for humans. And so you can see, you would have a drift, if you can see my hands going like that. Like if we had an actogram like mice do, we would drift if we lived in a cave, and then we'd cycle back around to the rhythms of people who are living outside with light. And so that will actually happen and can be an issue in people who don't have light perception. Yeah.
Wow,
it's
fantastic.
Really cool.
Challenging studies to do, I would imagine. Recruitment
Yes.
might be a little bit tricky for those
Yeah.
kinds of things. A question here, there's a few on this kind of topic, but how long does it take to recover from a night of sleep deprivation? I guess how long do the effects, the reporting insulin sensitivity changes persist, and this sort of thing?
It's such a good question. So it probably depends on what is the outcome. So I'll answer with respect to insulin sensitivity, but very few studies have actually looked at how much time it takes to recover. And also they're a little, they're variable, right? Because maybe one study gave, you know, ad lib sleep, and one study regulated sleep. So we did a sleep study where we gave 2 nights of recovery sleep to kind of mimic a weekend, and the participants averaged 10 hours of sleep. So if you think about what needs to happen for you to get 10 hours of sleep on average for 2 nights in a row, that was associated with a return to baseline insulin sensitivity. So that's one study. A different study that also provided 2 nights of insufficient sleep didn't see a full return to baseline insulin sensitivity. There's a lot of methodological differences that probably explain that, but I just want to say that, you know, 2 studies showed very different results, so we really don't know how long it takes to go back to baseline. But that would be cool, like a daily IVGTT.
Right. So instead of the 1 week, maybe multiple weeks in the room.
Yeah.
With, as someone pointed out, possibly IKEA furniture.
Yes.
So a question on the animal model. What questions can rodent models address to understand the relationship between sleep and metabolism? You've used the dog here, and you know the question is there enough translational relevance? And and maybe how would you define a good animal model of sleep?
Yeah. Oh, that's tough, because, you know, I would think about, do they have sleep architecture similar to us? Again, it really would depend on the exact question. You know, if I was wondering, the importance of slow wave sleep, then obviously the model, you'd have to, ensure that they are, that they have non-REM sleep, and is it comparable? But there's definitely, obviously there's many strengths to the rodent models, and especially tissue specificity. I mentioned that I'm not taking liver biopsies from healthy young individuals as much as I would love to, but that would be like a great, use of the animal model if we're really interested in changes to the liver in response to sleep and circadian disruption. The nice thing now too is that there's many more translatable methods of sleep loss in rodents, that they're, I showed that study with disc over water, which is like hugely stressful, very, not really translatable to the human condition of sleep loss, which is like, I stayed up too late watching The Office episodes, and it wasn't stressful. It was very relaxing, in fact. So they now have much more translatable methods like this, like slow moving bar that the animal has to move over, but it's not stressful, or, a little bit in the old days when all the grad students did gentle handling, which is basically how I kept the dogs awake.
Right. So you mentioned so it gives you an opportunity to test things you may not be able to do readily clinically. There's a question here on, does the location of the fat sample make a difference, visceral, peripheral, in the study?
Oh yeah, for sure. In humans, it's fairly difficult to get in healthy humans, like anything other than the sub-Q, although even sub-Q location will probably differ, whether it's depending where it is. But if you are at an institution where you could get visceral fat, for example, from gastric bypass surgery or other types of surgeries. So yeah, definitely the fat location would play a big role, and in just the function of the fat. So that's another place where an animal model might be ideal.
Okay, great. Interesting question here in relation to sleep disruption. So if you had a normal amount of sleep, the recommended amount 7 to 8 hours, but you had a disruption in the middle, so I suppose a transient interruption, does that affect insulin sensitivity?
I'm like, you're describing my life. Yeah. Every night. So yeah, unfortunately, yes, you can match sleep duration, but have reduced quality and see very comparable impairments. And we actually did that also at the University of Chicago, and Dr. Esra Tassali has really optimized and perfected this method where someone actually watches the EEG, and as soon as you start to see these big slow waves come, you can send an auditory tone to disrupt the participant into a lighter stage of sleep without actually waking them up fully. And so when we do that, duration doesn't change, but we can make the sleep a lot lighter, which is also potentially comparable to aging and some sleep disorders. We see an impairment in insulin sensitivity as well. So yes, sleep quality is very, very important, though in my opinion is difficult to study.
Right.
Sleep deprivation is like a sledgehammer.
Right. How would you evaluate typically sleep quality?
We use an EEG overnight.
EEG is the standard for it.
Yeah, that's for sure the gold standard. There's actually, there's a lot of research now into, wearable devices, and maybe we can start to get at some other aspects of sleep with skin temperature maybe, and heart rate, things like that.
Right. I think possibly we maybe need to wrap things up here.
Okay.
We'll forward all the questions on to you. Any last guidance for us in terms of sleep, what we should strive for?
Yeah, again, like I said, I'm not the best. I'm a naughty sleep researcher that watches The Office before bed, but limiting, we can, if you Google American Academy of Sleep Medicine guidelines, you can see a lot of tips there. But really, light at night and making sure that your bedroom is just for sleep, things like that are kind of the top tips. And just regularity. People are just starting to get some understanding that consistent sleep schedule is also probably a big player in this, not having a highly variable sleep schedule.
Right. Okay. Well, thank you so much for all your insights today, Josie. It's been a pleasure.
Sure. Yeah. Sorry about the Wi-Fi. That's going to keep me up. That's what I'm going to wake up at 3 a.m, cursing my name for that. Sorry.
Thank you. No problem at all. Thank you so much.
Thank you. Bye, Andrew.
Bye.
Bye, everyone.
Another quick thank you to our partners for helping make this series possible. And in closing, we hope you enjoyed this Inside Scientific webinar, and we'll see you again next time. Thanks, everyone.